Hello everyone, this is Vishal Raj from 2nd MSc Zoology. It is my video presentation about fertilization in mammals. Yes, before entering our topic, first of all, what is fertilization? A new life begins at fertilization, that is the union of two gametes, the egg and the spermatozoon. In mammals, it is very difficult to study. First reason is that fertilization in mammals is internal, occur inside the oviducts of the female. Why it is difficult to study? Mammalian fertilization is that the sperm population ejaculated into the female is probably heterogeneous, containing sperm at different stages of maturation. Out of the million of human sperm normally ejaculated during coitus. Coitus is sexual activity between individuals. Only 200 reaches the vicinity of the egg. It is difficult to assay the sperm to swim towards the egg and become activated. Site of fertilization in nearly all mammals is the ampulla, a wide part of the fallopian tube or oviduct. After fertilization in the ampulla, the egg are more correctly called as zygote. That is, the union of egg and sperm is called zygote. Approach of gametes. The precise place of oviduct where fertilization takes place in mammals is the ampulla. During copulation, male deposit their sperm in the upper vagina uterus, while oocytes are released from ovary. Both gametes use biochemical guidance as well as physical population to reach the ampulla. During transport of gametes to the final meeting place, oviduct plays very important active role. This process is called translocation. Transport of egg. The just released mammalian oocyte is surrounded by corona radiata. Preceding ovulation, hormonal changes result in greater muscular activity to the fimbaria of ostium, which increase the chance that oocyte will enter the oviduct. Once within the oviduct, the oocyte cumulus mass is driven downwards towards the ampulla by the powerful ciliary currents within oviduct. In human, oocyte takes about 3 days to reach the ampulla after ovulation. A released ovum from ovary transported to the ampullary stomach junction, that is ampulla. Transport of sperm. Sperm occurs in both the male reproductive tract and the female reproductive tract. In the male reproductive tract, transport of spermatozoa is closely connected with the structural and functional maturation, whereas in the female reproductive tract, it is important for spermatozoa to pass vagina, cervix, uterus to the fallopian tube, where they can meet the ovulated egg. After spermogenesis, in the seminiferous tubule, the spermatozoa are morphologically mature, but are non-motile and incapable of fertilizing an egg. Spermatozoa spend 10 to 12 days in the highly convoluted duct of the epidermis of human, during which time they undergo biochemical maturation. In majority mammals, including human, sperm are deposited in the upper vagina during copulation, but insemination in rodents occurs in uterus. The deposited sperm in the upper vagina pass through a cervix and pass through uterus, then reach the fallopian tubule. Sperm motility. Motility is nothing but flagellal action. Is probably important in getting sperm through the cervical mucus and into the uterus. Interestingly, in those mammals where the female is promiscuous, sperm from the same male will often from trains or aggregates where the combined propulsion of the flagella make the sperm faster. This strategy probably evolved from competition between males. In those species without such female promiscuity, the sperm usually remain individual. Uterine muscle contraction. Muscular contraction in the uterus are important for entry of sperms from upper vagina to the oviduct. Sperm's own motility due to flagellar activity is important once the sperm reach the oviduct. Directional guidance received by the sperm is provided by the temperature gradients between various regions of oviduct and by the chemical from oocyte or cumulus. On the journey from the site of insemination to the site of fertilization, a sperm attains maturity and capacity to fertilize an egg. Capacitation of sperms. A sperm just entered in the reproductive tract of female is unable to fertilize an egg. To attain the capacity of fertilizing an egg, a sperm has to reside inside the reproductive tract of female for some time, during which is subjected to poorly known influence of female tissue. The phenomenon by which a sperm becomes competent to fertilize an egg is called capacitation. Experimental proof. Plasma membrane of the sperm changed. The cholesterol in plasma membrane is removed by the albumin in protein of female reproductive tract. Cholesterol removal changes the location of region containing receptor protein on sperm cell membrane. That is, a fresh sperm has those regions throughout its plasma membrane, but after cholesterol removal, they are present on the sperm yet only. Capacitation results losing some glycoprotein covering on the sperm surface, probably removal of glycoprotein result in unmasking of the recognition sites of the sperm proteins which bind to zona pellucida of the egg. Phosphorylation of proteins in sperm yet result in forming the receptors, those bind to zona pellucida. Potassium ions leaves the sperm and membrane, potential of sperm plasma membrane becomes negative. As a result, calcium channels are opened and Ca++ enters the sperm. Such changes facilitates the membrane fusion even during acrosome reaction. Outermost membrane of acrosome vessel changes and comes in contact with the plasma membrane of the sperm. It seems to be a preparation for fusion of outer acrosome membrane with the plasma membrane during acrosome reaction. Contact of gametes. Capacitated sperms reaching ampulla have to penetrate the cell of corona radiata first and then zona pellucida to make contact with plasma membrane of the egg. Sperm penetrate through corona by their own swimming movement. Hyaluronidase, an enzyme on the outside of plasma membrane, facilitates sperm to digest path through the extracellular matrix of cumulus cell. Binding to penetrate through zona. Studies in mouse. 3 major glycoproteins. Zona proteins are called ZP. ZP1, ZP2 and ZP3 are found in zona pellucida of mouse. The zona proteins synthesized by maturing oocyte, crucial in binding the sperm then to start acrosome reaction. Acrosome reaction and penetration of sperm through zona. As soon as ZP3 cross-links with the receptor on sperm cell membrane, acrosome reaction is induced. One protein on sperm cell surface is galactosyl transferase, that is called as GALT. Active site of GALT is directed outward and binds N-acetyl glucosamine, the carbohydrate moieties of ZP3. Following this cross-link, specific G protein in the sperm cell membrane are activated, and a cascade of changes occur to open calcium channel on the sperm plasma membrane. The ultimate result is calcium exocytosis of the acrosomal vessel. A number of proteases are released from the exocytosis of acrosomal vessel. These enzymes make a hole in zona pellucida through which the sperm proceeds toward the egg. During acrosome reaction, anterior portion of cell membrane shed from sperm, but the sperm should retain some addition to zona. If it is going to penetrate it, it is recognized in the mice that proteins in the inner acrosomal membrane bind specifically to carbohydrate dishes of ZP2 and maintain The sperm zona attachment. Sperm having acrosome reaction completed can transfer the binding from ZP3 to the adjacent ZP3 glycoprotein. A trypsin-like enzyme called acrosine acts to produce this passage. This enzyme is bound to the inner membrane of acrosome and becomes exposed to the surface of sperm. Yet, after the acrosome reaction, surely proper action of sperm flagella and with the enzymatic digestion to penetrate the zona pellucida by the sperm. Capacitated sperms, after reaching ampulla, have to penetrate the cells of corona radiata. After crossing the corona radiata, the acrosomal reaction takes place in zona pellucida and the sperm penetrates through zona. Union of sperm and egg. Union of gamete is facilitated by chemicals from both gametes and microvilli projected from the egg. Mammalian sperm touches the side of the egg, not at the tip of it. Fusion of sperm cell membrane with that the egg begins at the junction between acrosomal membrane and sperm cell membrane, called eukaryotic region. Where the eukaryotic region touches the egg, microvilli are extended from the egg membrane due to polymerization of actin. The gametic fusion in mammals depends on the interaction between sperm protein and egg protein. One sperm protein associated with gametic fusion is isomo. This is nothing but an immunoglobulin-like protein. From egg site, CD9, an integrin associated protein, is responsible for gametic fusion. Errors of fertilization. Polyspermy. Polyspermy is fertilization by more than one spermatozoa. It occurs naturally in few cases and can be enhanced by aging of the egg before fertilization.